Okay, so welcome back to this next video in which we are discussing Cys-Loop receptors. Okay, right. So in the previous video what we discussed is the basic structural features of all Cys-Loop ligand-gated ion channels. Okay, so we discussed that the full Cys-Loop ligand-gated ion channel is a pentamer made up of 5 separate subunits joined together. Okay, and if you pull out one of these subunits of a Cys-Loop ligand-gated ion channel and have a look at its basic structural features, we can reduce it down to what we see here. Okay, so the N-terminus of the Cys-Loop ligand-gated ion channel subunit is extracellularly, okay, and one of the key domains that you have extracellularly is this cis-loop here, okay, this loop in the polypeptide structure held together by a disulfide bond between 2 cysteine residues, okay. Then following the extracellular domain you have the 4 transmembrane alpha helices, okay, M1 to M4 here, with this intracellular loop between the M3 and M4 membrane-spanning alpha helices. Okay, right. So what I want to do at the start of this video then is discuss different examples of Cys-Loop ligand-gated ion channels and the ions that they are permeable to, and also examples of where they are physiologically important. Now note, we're going to come back to most of these examples later on and discuss them in more detail once we've discussed the in-depth structure of a Cys-Loop ligand-gated ion channel and the opening mechanism. Okay, but for now it's a basic introduction to the different types of Cys-Loop ligand-gated ion channels. Okay, so we're firstly going to start off then with the mammalian families of Cys-Loop ligand-gated ion channels. Okay, so the first one that I want to then discuss is perhaps the most famous of all the Cys-Loop ligand-gated ion channels, which are the nicotinic acetylcholine receptors, which I will abbreviate down to NACHRs, like so. So this stands for nicotinic, that's the little N here. ACH then stands for acetyl, that's the A, okay, and then the CH is for choline, okay, and then the R is then for receptors, so nicotinic acetylcholine receptors. Okay, so firstly, let me discuss the neurotransmitter acetylcholine then I'll discuss examples of where nicotinic acetylcholine receptors are incredibly important in physiology, okay, and we'll discuss what ions they are actually permeable to as well. Okay, so for now let's discuss acetylcholine then. So basically this is an acetic acid molecule esterified to a choline alcohol molecule. Okay, so let me show you the acetic acid group. Okay, so acetyl refers to the acetic acid group, so acetic acid is just a 2-carbon carboxylic acid molecule, okay, like so. So here is the methyl group and here is the carboxylic acid group. And now this is going to be esterified onto the alcohol group of a choline molecule. Okay, so what is choline? Well, choline is a molecule where you have an alcohol group at one end, which we've esterified onto the carboxylic acid group of an acetic acid molecule. Then what you have is an ethylene group, okay, which is just 2 methylene groups joined together. So 2 carbon atoms, each with 2 hydrogens coming off, okay, which I'll show like so. And then at the other end, what you're then going to have is a quaternary ammonium group, okay, so you're going to have a nitrogen atom here, and it's going to have 3 methyl groups coming off it. Okay, and that overall gives this nitrogen atom here in the choline molecule 4 bonds coming off it. Now, nitrogen's not supposed to have 4 bonds coming off it. The ideal number of bonds for a nitrogen atom to have coming off it is 3, not 4. Okay, if nitrogen has 4 bonds coming off it, what it means is that one of these bonds was not a fair bond, basically, okay, because usually covalent bonds, the deal is that one atom puts in 1 electron and the other atom puts in another electron, okay, and that's the 2 electrons in the covalent bond, okay. In one of these bonds, and let's this one at the top here, the nitrogen will put in both electrons, like so, okay, so usually when nitrogen just has 3 bonds, okay, so let's imagine it just had these 3, it would then have a lone pair of electrons here. but basically now it's formed 4 bonds. This lone pair of electrons has actually formed the covalent bond, okay, with no contribution at all from this carbon atom here. Okay, now because the normal understanding of a covalent bond is that one electron belongs to each of the constituents of the bond, then the understanding is that one of these electrons belongs to the nitrogen now and the other belongs to the carbon. So basically the nitrogen has given away an electron to the carbon, and that gives this nitrogen here a positive charge. Okay, like so. Right. Now you might ask, well, doesn't the carbon now have a negative charge because it's received an electron from the nitrogen? And the answer is no, because when the carbon entered this sorted arrangement here, it will come in with a positive charge basically originally, okay? And then when it received an electron from the nitrogen, that neutralized its positive charge and effectively it deferred the positive charge onto the nitrogen atom. So that means that this choline molecule actually has a positive charge here, and that will become important when we want to discuss how acetylcholine actually binds to the acetylcholine receptor. The positive charge is going to be extremely important in the example of the nicotinic acetylcholine receptor ligand binding pocket. Okay, right, so that then is the neurotransmitter acetylcholine, and just to stress again, this is the choline molecule here. Okay, so if you broke off this acetyl group here and just put a hydrogen off this oxygen, that would be a choline alcohol molecule. Okay, acetylcholine is then the acetic acid molecule attached to the choline molecule. Okay, and for short, acetylcholine then is often just abbreviated to ACH, like so. Okay, right, so let's now talk about physiologically important examples of where acetylcholine is used as a neurotransmitter. Okay, so obviously the most famous example is the neuromuscular junctions, the NMJs. Okay, so this stands for neuro, that's the N, and then muscular, that's the M, and then junction. Okay, so these aren't counted as synapses, but it's a very similar concept to a synapse. Okay, what you've got then is an axon terminal from an alpha motor neuron synapsing on your skeletal myocyte here. Okay, so I'll draw this here. So remember, skeletal myocytes are massive great cells. They're really syncytiums of multiple cells joined together, okay, into one great massive supercell. Okay, and basically, if you want to make the skeletal myocyte contract, this axon terminal of this alpha motor neuron releases acetylcholine onto the skeletal myocyte, and then the acetylcholine is going to work on nicotinic acetylcholine receptors on the surface of the skeletal myocyte and open ligand-gated ion channels then, which will allow an excitatory current into the cell. Okay, so let's just discuss what the current is in the case of nicotinic acetylcholine receptors. So what are nicotinic acetylcholine receptors, this type of cis-loop ligand-gated ion channel, actually permeate to? Okay, well, they allow sodium ions and also potassium ions to move through them. However, the most important one to actually know about in this case is the sodium ions rather than the potassium ions. Now, why is that? Okay, well, remember that before we've actually triggered an action potential in the skeletal myocyte, the electrical potential difference across the cell membrane of the skeletal myocyte will be very much so negative. In fact, the electrical potential difference across skeletal myocytes is very extreme, okay, much more so than across a normal neuron. In fact, I think it's something like negative 90 millivolts is the electrical potential difference across the cell membrane of a skeletal myocyte. Okay, so that means that the intracellular electrical potential is 90 millivolts lower than the electrical potential extracellularly. Okay, so if we now think about what the effect of opening an ion channel that is permeable to sodium and potassium ions is going to be on these skeletal myocytes. Okay, here is our nicotinic acetylcholine receptor, which is this type of Cys-Loop ligangated ion channel, so it will have all of those basic structural features that we've just been talking about. Okay, and it's been opened by the binding of acetylcholine molecules onto the surface of the nicotinic acetylcholine receptor, and now it's going to be permeable to sodium and potassium ions. What sort of currents are we going to get? Well, we know the electrical potential difference across the cell membrane, but we also need to know the concentration gradients for sodium and potassium ions across the cell membrane if we're going to work this out. Okay, so usually sodium concentration extracellularly is around 145 millimolar, whilst intracellularly sodium concentration is 12 millimolar. Okay, so that's 12-fold gradients of sodium favouring its movement into the cell. Okay, we also know that the electrical potential difference across the cell membrane is around negative 90 millivolts, meaning that the electrical potential intracellularly is 90 millivolts lower than the electrical potential extracellularly. Okay, so that means that both the concentration gradient and the electrical gradient are both telling the sodium ions to go into the cell, basically. Okay, they're both in agreement in this case, because sodium wants to go where the electrical potential is lower, which in this case is the intracellular compartment here by 90 millivolts. Okay, so sodium ions are going to come in basically when you open this channel. Now for potassium ions, it's much more difficult to decide. Okay, potassium ions, the extracellular concentration is around 4 millimolar, and the intracellular concentration is around 155 millimolar. Okay, so that's a 39 or 40-fold gradient of potassium favouring its movement out of the cell. But then potassium ions are also a positively charged ion, and therefore want to go when the electrical potential is lower. So this one's much more difficult to decide, because the concentration gradient says go out, the electrical gradient says go in. They're saying the opposite thing. The question now is who wins? Okay, well, basically, at around negative 90 millivolts, this is actually stronger than the concentration gradient, so you might get a tiny movement in. The Nernst potential for potassium is around negative 85 millivolts. Okay, the Nernst potential is the electrical potential difference that you need across the cell to completely stop any movement of potassium. Okay, so if the electrical potential difference was negative 85 millivolts, then the electrical gradient would be completely balanced with the concentration gradient, and you get no movement of potassium. Okay, we are actually more hyperpolarized than the Nernst potential for potassium, and therefore you're expecting now the electrical gradient to win and to bring a tiny little movement of potassium into the cell, but tiny. Okay, and as soon as the electrical potential difference across the cell membrane goes depolarized from negative 85 millivolts, let's say down to negative 80 millivolts, then it will reverse and you'll get potassium moving out now, because the concentration gradient will then beat the electrical gradient. But the point is here, the potassium movement is tiny compared to the sodium movement, so neglect the fact that these nicotinic acetylcholine receptors are permeable to potassium. You might as well forget it. It's an important thing to know, okay, but you might as well forget it. The sodium gradient and the electrical gradient acting on the sodium ions is what's important, okay, because they are going to actually create a very large sodium current coming into the cell here, and that then is going to depolarize the electrical potential difference across the cell membrane. Okay, so nicotinic acetylcholine receptors are permeable to both sodium and potassium ions. However, in physiologically important terms, it's only the sodium current that's important, because normally the electrical gradient across cell membranes is going to be opposing the concentration gradient of the potassium ions, okay, whereas it's going to be working alongside the concentration gradient of the sodium ions. And therefore you're going to get large sodium currents coming into the cell, and then you'll get tiny little potassium currents either going out of the cell or coming into the cell, depending on which side of the Nernst potential for potassium you're on. Okay, but we will neglect the potassium currents basically. We're just going to focus on the sodium currents. And indeed, we're going to see many other examples of Cys-Loop ligangated ion channels that are permeable to both sodium and potassium ions. And again, we will just sort of dwell on the permeability to sodium ions and the fact that that is going to cause depolarization, because that's the main current that's actually going to move through these channels when you open them. Okay, right. That's one of the very important examples then of where nicotinic acetylcholine receptors are found on the skeletal myocytes, and they're responding to acetylcholine released by alpha motor neuron axon terminals. Okay, and they will cause the depolarization of the electrical potential difference across the cell membrane of the skeletal myocyte, and therefore make it more likely that the skeletal myocyte will fire an action potential, which will then cause it to contract. Okay, right, some other important examples, however, of nicotinic acetylcholine receptors. They are also at the autonomic ganglia, okay? So let me just explain what the autonomic ganglia are. So basically, in the autonomic nervous system, you have these two branches, the sympathetic nervous system and the parasympathetic nervous system, okay? And generally, they affect the activity of internal viscera. So for instance, the heart, the liver, the adrenal glands, things like that, okay? Now, when the autonomic nervous system wants to actually synapse onto an internal viscera, generally what happens is you have an autonomic neuron that has its cell body inside the spinal cord, comes out of the spinal cord, okay? But then it isn't the neuron that actually synapses onto the viscera. Instead, what you have is then a cell body of a second neuron that's outside of the spinal cord, which will then be the neuron which synapses onto the target, basically. Okay, so this is the way the autonomic nervous system is usually organized. Okay, here is the neuron with its cell body inside the spinal cord. So I'll show the spinal cord here. Okay, its axon will then leave the central nervous system and then synapse onto a second neuron which has its cell body outside of the spinal cord, and that's that second neuron which then will synapse onto the actual target of the autonomic nervous system. And this is for both sympathetic and parasympathetic neurons. Okay, right. So basically, when you have cell bodies of neurons outside of the central nervous system, that is called a ganglion. Okay, and these autonomic neurons that are prior to the ganglion, these are called the preganglionic neurons. Okay, so this is called the preganglionic neuron. And then the neuron that is after the autonomic ganglion, this is called the postganglionic neuron, and this is the one that actually synapses onto the target. Okay, and basically the preganglionic neurons of both sympathetic nervous system neurons and parasympathetic nervous system neurons use the neurotransmitter acetylcholine to stimulate the postganglionic neuron. Okay, so acetylcholine is released by this preganglionic neuron onto the postganglionic neuron, and it will then work on nicotinic acetylcholine receptors on the dendrites of the postganglionic neuron to try and make this postganglionic neuron then fire. Okay, and again, these nicotinic acetylcholine receptors will opening the sodium and potassium conductive channel, but again, the most important current is the sodium current coming in, which will depolarize the electrical potential difference across the cell membrane and make it more likely that this postganglionic neuron will fire an action potential. Okay, so I'll reiterate that this is for both the sympathetic and the parasympathetic nervous systems. I haven't specified whether these preganglionic and postganglionic neurons are sympathetic or parasympathetic. It's the same for both. You have the cell body in the central nervous system here of the preganglionic neuron. It will send its axon out into the sympathetic or parasympathetic ganglion here, and then where it will synapse then onto the postganglionic neuron here, and that synapse will involve nicotinic acetylcholine receptors and the release of acetylcholine by that preganglionic neuron here. Okay, right, so that's another physiologically important example of where nicotinic acetylcholine receptors are going to be involved. Okay. Finally, I should also say that you do have some nicotinic acetylcholine receptors which are in the brain. Okay, so acetylcholine is a neurotransmitter which can be used as an excitatory neurotransmitter in the brain. Okay, so it's also in the CNS, and we will look at examples of CNS nicotinic acetylcholine receptors later on, and it's used as an excitatory neurotransmitter in the CNS. It's not the major excitatory neurotransmitter in the brain. The major excitatory neurotransmitter in the brain is glutamate, okay, but the glutamate receptors are not cis-loop ligand-gated ion channels. They are ligand-gated ion channels, but they have completely different structures to the cis-loop ligand-gated ion channels. Specifically, they're not pentamers, they are tetramers. Okay, so they're not within this same family that we are now in the process of discussing. Okay, right, so that's the nicotinic acetylcholine receptors. Now let's turn our attention onto another family of cis-loop ligand-gated ion channels. And the next family of cis-loop ligand-gated ion channels that I want to discuss are the GABA A receptors. Okay, right. So I've just said that the major excitatory neurotransmitter in the brain, okay, the major neurotransmitter that's released onto neurons in the brain and which increases the probability of that neuron firing an action potential by opening ligand-gated ion channels which allow excitatory sodium currents into the cell, is glutamate. Okay, the major inhibitory neurotransmitter in the brain, which when released onto neurons makes it less likely that that neuron is going to fire an action potential, and it does this by opening ligand-gated ion channels which allow inhibitory chloride currents into the cell, is GABA, okay? So let's just discuss what GABA is, and these GABA A receptors are the main receptors which are chloride ligand-gated ion channels, basically, which are going to inhibit neurons, basically, when they're open from firing action potentials. Okay, so let's now discuss then this neurotransmitter GABA, okay, and then we'll discuss the GABA A receptors. Okay, so firstly then, what does GABA actually stand for? Well, it stands for gamma aminobutyric acid. So the G is for gamma, okay? Then the A is for amino, the B is then for butyric, and the final A, the final A is for acid. So forget that little line there, it should be just gamma aminobutyric acid. Okay, so that's usually just abbreviated then down to GABA. Okay, right, so let's have a look at the structure of GABA. So it's a very simple molecule. Okay, all it literally is butyric acid with an amino group added onto it. Okay, so what is butyric acid here? Okay, well, butyric acid is the old name for a molecule that would now be called butanoic acid. Okay, so the new name for butyric acid is butanoic acid. And this, therefore, is just a 4-carbon carboxylic acid molecule. Okay, so let me show this here. So here is the carboxylic acid group here. Okay, now all we need is an additional 3 carbons to take this molecule up to a total of 4 carbons. Okay, now before I add any hydrogens on there, let's just discuss the gamma amino, because we don't just want butyric acid, we want gamma aminobutyric acid. So we need to add an amino group on to one of these carbons, but which carbon is it? Well, it's the gamma carbon. Okay, what does gamma carbon mean? Okay, well, this is the old way of naming the carbons within a carboxylic acid molecule. Okay, so the new way of naming carbons in a carboxylic acid molecule is just to call this one number 1, this one number 2, this one number 3, and this one number 4. But the old way of naming the carbons in a carboxylic acid molecule is to start with the one that comes off the carboxylic acid group. Call that the alpha carbon. Okay, alpha is the first letter of the Greek alphabet. Then the second one here, you'd call that the beta carbon, second letter of the Greek alphabet. And then this one here would then be the gamma carbon. So we want an amino group coming off this gamma carbon here. Okay, and that will create as gamma aminobutyric acid. So now let me just finish the molecule off by sticking hydrogens off all the other places here. Okay, so just 2 more hydrogens to go. Right, so that's the gamma aminobutyric acid molecule. That is GABA, the major inhibitory neurotransmitter in the brain. So let me repeat this. If a neuron wants to inhibit another neuron, okay, so we talked about this right at the beginning, neurons have a huge number of other neurons synapsing onto them. Okay, and some of these neurons will be trying to excite them, and other neurons will be trying to inhibit them. The major excitatory neurotransmitter that neurons can release onto each other is glutamate. Okay, so if you want to have a synapse which is an excitatory synapse, then the presynaptic neuron will release glutamate onto the postsynaptic neuron, which will then have ligand-gated ion channels for glutamate, which we're not going to discuss in this video because they're not cis-loop ligand-gated ion channels. Those will then open and allow in sodium ions into the cell. Okay, if on the other hand you want to have an inhibitory synapse, okay, what you will have is the presynaptic neuron will release GABA onto the postsynaptic neuron. At least usually, these are the main neurotransmitters. Not all neurons use these neurotransmitters. We've discussed that acetylcholine is occasionally used, okay, but compared to glutamate, acetylcholine is hardly ever used as a stimulatory neurotransmitter, okay. So going back to GABA, the presynaptic neuron will release GABA onto the postsynaptic neuron, which will then have ligand-gated ion channels for GABA on its surface, which will be of this type, cis-loop ligand-gated ion channels. And when they open, they will allow chloride anions into the cytoplasm of the dendritic spine here, and those chloride anions will go to the axon hillock and reduce the probability that the axon hillock will fire an action potential. Okay, right, so we now want to have a little bit more of a discussion of the GABA A receptors then. So basically, in the brain you have two major types of receptors for this neurotransmitter GABA. You have the GABA A receptors, okay, which are cis-loop ligand-gated ion channels, and then you have another class of GABA receptors, which are known as the GABA B receptors, which are not cis-loop ligand-gated ion channels. In fact, they're not even ligand-gated ion channels. These are, in fact, G-protein coupled receptors for GABA. Okay, now the main way that GABA works on other neurons is through GABA A receptors. These are the ones which can deliver this inhibitory chloride current into the cytoplasm of postsynaptic neurons. Okay, so most GABA receptors will be GABA A receptors. GABA B receptors are generally presynaptically located and are involved in presynaptic inhibition of the presynaptic axon terminal. Okay, right, so that's all I want to then say about GABA receptors for now. Okay, they are a chloride channel, basically, once they're open, and therefore they inhibit postsynaptic neurons. Okay, they make it less likely that postsynaptic neurons are going to fire an action potential. And it's a major, major neurotransmitter within the brain. We will come back to these receptors later on and discuss all the different types of subunits that you can use to build them and the fact that they are entire families. So I should stress that now, actually. Nicotinic acetylcholine receptors and GABA A receptors and the 5-HT3 receptors and the glycine receptors that we're about to discuss, they are not just one receptor, okay, they are entire families of receptors that are all in this superfamily of cis-loop ligand-gated ion channels. Okay, so all cis-loop ligand-gated ion channels have these core structural features that I've shown you. They have this pentameric nature where you have 5 subunits joined together to make the full cis-loop ligand-gated ion channel. The cis-loop ligand-gated ion channel subunits have the membrane spanning topology that I've shown you. Okay, they're going to all have the more in detailed structural features that I'm going to tell you about later, but there's a huge amount of diversity amongst the cis-loop ligand-gated ion channels. There are these different families which all respond to very different ligands and which conduct very different ions when opened. Okay, and these groups that we are now discussing are not individual types of channel. Okay, they are whole families of channels. Okay, right, and we'll come back to that later, so if you don't understand that at the moment, it will become more obvious later. Okay, right, so I want to discuss another family of cis-loop ligand-gated ion channels that are very similar to the GABA A receptors and therefore should be put right after the GABA A receptors. Okay, and these are the glycine receptors, okay, known as the GlyRs. Okay, so this stands for glycine, that's the gly, and then just receptors. Okay, so Basically, GABA is a major inhibitory neurotransmitter used in the brain, and it's also used in the spinal cord. Okay. Glycine is another major inhibitory neurotransmitter that can be used, but it's not found really in the brain. It's found predominantly in the spinal cord. Okay, so first let me just show you the structure of glycine. So glycine is one of the 20 amino acids that's used in proteins, okay? And it's actually the simplest of the proteinergic amino acids. So let me just show you its structure. So here's the amino group, like so. Here's the alpha carbon with a hydrogen coming off it, so I'll just firstly show the core amino acid structures, and here's the carboxylic acid group, and then the R group of glycine is quite simply a hydrogen atom. Okay, so that there is the structure of glycine. Okay, and just like GABA, it can be used as an inhibitory neurotransmitter to decrease the chance that the post-synaptic neuron is going to fire an action potential. Okay, and it's used by inhibitory interneurons in the spinal cord, and when we come back to discuss these in more detail, I'll discuss examples of what inhibitory interneurons might be inhibiting. Okay, specifically they can inhibit alpha motor neurons, and that's going to be important in hyperekplexia. Okay, so again, when they open, in response to the glycine binding, they open a chloride channel, and that's then how they can deliver this inhibitory current to the postsynaptic cell. Okay, and therefore make it less likely that the postsynaptic cell is going to fire an action potential. Okay, right, so I think we'll have a break there. And in the next video, what we'll move on to is we'll continue discussing the different families of cis-loop ligand-gated ion channels. So we'll go on to the 5-HT3 receptors, the final family of receptors in mammals that are cis-loop ligand-gated ion channels. And then we'll look at these non-mammalian forms, so the glutamate-gated chloride channel and ilic and glic. And then what we'll do is discuss the structure of cis-loop ligand-gated ion channels in more detail.